The first and longest phase of mitosis is prophase, and that's when chromatin condenses into chromosomes. So that really loose stuff comes tighter and tighter into the chromosomes over here. And then the nuclear envelope and the nucleolus, they disintegrate. And so this allows the chromosomes to be in the cytoplasm, and also spindle fibers start forming. Spindle fibers are just structures that will be essential to moving the chromosomes in later stages of mitosis. And spindle fibers are made out of these things called microtubules. So, after prophase comes prometaphase, which is the second phase of mitosis, but it's often denoted as part of metaphase. So, sometimes you'll see prometaphase, sometimes you'll see metaphase. And here in prometaphase, the spindle fibers attach to the centromeres. The centromeres are just the middle of the chromosomes. And between the chromosomes, the centromeres, and the spindle fibers, there are these protein complexes called kinetochores that help the spindle fibers attach to the centromeres. And then the spindle fibers start pulling, like they start moving the chromosomes. And that brings us to metaphase, which is the third phase of mitosis. And basically, first we have our metaphase checkpoint. And here, proteins check whether all of the chromosomes are attached to spindle fibers or not. And this is the last checkpoint before, for the cell cycle. So we had our G1 checkpoint, G2 checkpoint, and our last checkpoint is the metaphase checkpoint. And then hopefully everything goes well from there because there are no more checkpoints. So after the metaphase checkpoint, basically metaphase happens, and that's when the chromosomes align at the metaphase plate, which is just a line down the middle of the cell, an imaginary line, kind of like the equator. And basically, the spindle fibers are the ones who move the chromosomes to the metaphase plate. After metaphase, we get anaphase. And in anaphase, we have, that's the fourth phase of mitosis. And this is where the spindle fibers pull apart sister chromatids. So basically before we had this kind of an X-shape chromosomes. But now each of the strands of those X are pulled apart. And each of the strands were called sister chromatids, but now because they're separate, they're kind of fully functional now, and they're called daughter chromosomes. And here, telophase. Before we get into telophase, just one clarification. Earlier we mentioned that the chromosomes, you got one from mom, one from dad. That's kind of not correct, because basically each one of these sister chromatids are identical. And part of the sister chromatids come from mom, and part of it comes from your dad. So each of them is kind of a mix between the two parents' chromosomes. But just to clarify, these daughter chromosomes are identical, and in fact mitosis is basically just taking a parent cell and dividing it into two identical daughter cells. So now we have telophase, our fifth and final stage of mitosis, and this is where the spindle fibers disintegrate, the daughter chromosomes diffuse into chromatin, so once again these spaghetti strands, and the two nuclear envelopes and the two nucleoli reform. So it's kind of like a reverse prophase kind of thing, except you have two nucleuses. So mitosis